This is a podcast intended for IB biology students studying for their exam. We're going to be going over Chapter 2, which is cells, and more specifically sections 2.2 and 2.3, which is prokaryotic cells and eukaryotic cells. We'll specifically be going over individual assessment statements, and section 2.2 has 4, I believe, and section 2.3 has 6. Okay, so we're going to start off with section 2.2, which is prokaryotic cells. The first assessment statement asks for you to draw an E. coli as an example of a prokaryote. So the first step is to draw the body. Once you have done that, add 3 layers to your body. I usually make this one a little thicker, like this. Okay, so what do we have here? The first layer is called a slime capsule, which is used for protection against desiccation or phagocytosis. The second layer, this one right here, is a cell wall, which gives the cell shape and helps in protection. The third one, the one that I painted black, is the cell membrane. It regulates the flow of materials in and out of the cell. Next, what you're going to do is you're going to draw a nucleus or a nucleoid. So that can be just squirmy, and that's a nucleoid. Basically, this is all the genetic material inside the cell. Also inside the cell, draw little tiny spots and kind of like bigger things and more tiny spots. The small dots will be your ribosomes. Ribosomes is where protein synthesis occurs, and these kind of squiggly organisms here are your plasmids, which is basically additional DNA molecules that can exist and replicate themselves independently. Never forget the cytoplasm, which is just this whole area right here. The cytoplasm holds the organisms and the enzymes and the nucleoids together inside the cell. We have the pili on the outsides, which you can draw as like tiny little tails coming out of the cell. Their purpose is for cell-to-cell adhesion, attachment to solid surfaces, or it is used in the process of transferring DNA from one cell to the other. And I almost forgot the flagella, which is a tail coming out of the end of your cell, used for mobility. So that is it for the first assessment statement. And with that, we also complete the second one, which is to annotate the diagram and know the functions of each of these parts. The third assessment statement asks for you to identify electron micrographs of these structures in the E. coli. And the fourth assessment statement is to state that prokaryotic cells divide by binary fission. And what is binary fission? It's a type of asexual reproduction. And what happens is that first a replication signal is sent and the DNA is copied. Then these DNAs attach to the membrane of the cell, and it splits through cytokinesis, and that's it. But do not confuse this with mitosis. That's the end of section 2.2, and we will be moving on to eukaryotic cells next.
A eukaryotic cell is fairly different than a prokaryotic cell. We will later be comparing both of them. But an example of a eukaryotic cell here is an animal cell, or a liver cell, which is an example of an animal cell, specifically stated as stated in the assessment statement. So first, should you draw a eukaryotic cell? First, you should draw the cell membrane, and make sure to not draw a specific and rigid framework and more kind of a round shape cell, for the animal cell does not contain a cell wall and only a cell membrane that will control what comes in and out of the cell, as well as will separate what is from the inside and the cytoplasm and the organelles from what is the outside of the cell. A really important organelle is the nucleus. So you can name all of this organelle as a nucleus, which is composed of a nuclear envelope, which has openings and pores and controls what goes in and out of the nucleus. And the nucleus is also composed of a darker region named the nucleolus. And nucleolus specifically is where the subunits of the ribosomes are formed. And so ribosomes is another organelle which it's within the entire cell and the cytoplasm of the cell. Make sure you make sure you name the organelle, and there is no need to draw the subunit of the ribosomes since this is a fairly small image of the cell. An organelle that is really important are the endoplasmic reticulum. So first, the rough endoplasmic reticulum, which is very close to the nucleus. The rough endoplasmic reticulum is composed of flat vesicles connected with each other by the lumen, and it collects proteins and molecules synthesized by the ribosomes that are attached to it. So make sure you draw some ribosomes within the organelle. Then right next to the rough endoplasmic reticulum is the smooth endoplasmic reticulum, which is also tubular vesicles that are connected to each other by the lumen. But specifically the smooth endoplasmic reticulum, smooth ER and rough ER, specifically the smooth produces lipids, steroids, hormones, as well as break down toxins and stars calcium. Then another really important organelle is the Golgi apparatus. Golgi apparatus. The Golgi, differently from the endoplasmic reticulums, is composed of a pile of circular flat vesicles that are physically independent from each other, so make sure you draw some space within each vesicle. And the Golgi will then process all the lipids from the smooth ER and the proteins from the rough ER through vesicles that are going to move to the Golgi, and the Golgi will process everything that comes to it. Another really important organelle is the lysosome. The lysosome contains enzymes that will digest the external material that enters the cell, as well as the cell's own material that are either not needed anymore or they are not functioning properly within the cell. Another really important organelle is the mitochondria. So the mitochondria is composed of folding-like crystals. Make sure you draw that. Mitochondria is where the ATP synthesis occurs, and it has its own DNA. And the number of mitochondria in a cell depends on all the cell's need for ATP. There is no need to draw a chloroplast specifically, as this is an animal cell and not exactly a plant cell. There is also no need to draw vacuoles for the same reason, for no excess enough of water or nutrients is needed. But centrioles, which look like small little batteries that are perpendicular to each other near the nucleus, they are related to the production of the cytoskeleton, and then they are made of tubes called microtubule. Make sure you cannot forget to name the cytoplasm, which is the gel-like fluid that holds all of the organelles into place.
So the first assessment statement is comparing prokaryotic and eukaryotic cells. We have three that are very easy to remember. So first difference. Prokaryotic cells have DNA not in protein form, while eukaryotic cells have DNA in protein form. So these two. Then, in prokaryotic cells, the DNA is free in the cytoplasm, while in eukaryotic cells, it is located in the nucleus. And the third of them, in prokaryotic cells, there are no mitochondria, while in eukaryotic cells, there are mitochondria, and they work as the producers of energy, or ATP. Finally, in prokaryotic cells, the ribosomes are of the 70S kind. That's a designation for size. And in eukaryotic cells, the ribosomes are of the 80S kind, and that means that in eukaryotic cells, the ribosomes are bigger. And in addition, prokaryotic cells are of the size less than 10 micrometers, while eukaryotic cells tend to be bigger than 10 micrometers. The next assessment statement is comparing plant and animal cells. Plant cells have a cell wall, while animal cells do not. Plant cells have chloroplasts, while animal cells have no chloroplasts. Plant cells also have large vacuoles, usually centrally located, while animal cells have no or small vacuoles. Also, plant cells store energy in the form of carbohydrates as starch, while animal cells use glycogen. In addition, plant cells have no centrioles, while animal cells have centrioles. Finally, plant cells have a fixed angular shape because of the cell wall, while animal cells are more flexible and tend to have a round shape because they have no cell wall. Now, the two roles of the extracellular matrix. One of them is coordinating cell-to-cell interaction, which also leads to the coordination of some activities within the cell. Finally, some research indicates that factors in the extracellular matrix lead to cell migration and movement.